My name is Hannah Easley. I am a computer science major, a chemistry major and a computer science minor here at Oglethorpe University. And my research project has been conducted under the guidance of Dr. Allison Ressler. The name of my presentation is Investigating the Impact of Anion Pairing and Isomerization on the Energies of DCPD. So before we get started, some background information is definitely needed. We'll start with what is a polymer. So a polymer is a big molecule made up of smaller molecules. They're called monomers. So in this example, we can see that ethene would be the monomer, and then it is polymerized into polyethylene. If a polymer gets big enough, it can even be seen by the human eye. That is known as a macromolecule. Polymers are super common and we see them every single day in our lives. Some examples include rubber, plastic, and even DNA. These are all compounds that are made up of individual compounds bound together through polymerization. So one way to polymerize molecules is through a process known as ring-opening metastasis polymerization. ROMP is a way to polymerize specific monomers known as cyclic monomers. They're called cyclic monomers because they have a carbon ring inside of them. So what happens with ring-opening metastasis polymerization is that the double bond is broken and the monomer is open so they can join with other monomers. However, only one ring is opened and it is always broken at the double bond. So as we can see in this bicyclic monomer, the double bond is broken and this ring opens. However, the secondary ring is retained. This is also shown in the third example. So when we polymerize molecules, they can be added to one another in either a cis or a trans conformation. So these two molecules are the exact same representation, but from a different perspective. So when you join this monomer to another monomer, it can add in a cis or a trans conformation. Cis conformation means that the bonds are at the same side of the double bond. By contrast, a trans configuration would result in them added at opposite sides of the double bond. Because of how the monomers add to the growing chain as either cis or trans, it is possible to design a catalyst that can control the orientation of the added monomer. This would be an example of stereoselectivity. The reason we care about how a monomer is added as either cis or trans is because if you have a high proportion of cis or trans content, this can affect properties of the polymer, including melting point, tensile strength, and more. One example that you might be familiar with is a rigid solid plastic versus plastic film. This is an example of a macromolecular effect caused by the micromolecular difference. It is therefore desirable to design catalysts that can control the stereoselectivity so that they can be used in the construction of polymers. So ROMP, as I previously described, is a polymerization process. However, it relies on metal catalysts. So a catalyst is a compound that is added to a reaction in order to speed up the reaction, enable the reaction, etc. The use of a metal catalyst means that there will be a metal byproduct because the metal is not needed in the final product. Metal byproducts have to be disposed of, and as a result, they contribute to heavy metal poisoning, where typically they will leach into waterways, and when reabsorbed by vertebrates, they can cause central nervous system dysfunction and more issues. Therefore, a metal-free ROMP mechanism would minimize the environmental impact of this crucial industrial process. So the first metal-free ring-opening metastasis polymerization mechanism was reported in 2015 by Suijin Yang. She, in conjunction with the Boydston Research Group, were able to discover conditions that resulted in more cis or trans content, but they didn't have a full understanding of how to control the mechanism. So the cis and trans in this example would be shown by the position of the growing polymer arm. So the P represents where the compound joins to the other monomers to form the polymer, and You can therefore have 2 trans and 2 cis because the second arm of the opened ring can also assume different positions. So they were able to induce significant cis versus trans content, 75% cis and greater than 98% trans. And they hypothesized that the driving mechanism for this was in these intermediaries, meaning the cis versus trans nature of the intermediary would determine the ability to add as cis versus trans in the final product. This was accomplished by changing experimental parameters, but they didn't understand specifically why, and computational chemistry is how we can analyze the reasons why this mechanism works. So when a monomer is polymerized, meaning when the monomer is added to other monomers to form the polymer, it is a positively charged species. Therefore, it needs a negatively charged anion to stabilize it in the solution. The computations performed by the Boydson Research Group determined that there were steric reasons behind the cis-trans selectivity when considering the anion pairing effect. So they determined that a small anion pairing would result in trans selectivity and that a large anion pairing would result in cis selectivity. My research is from a computational chemistry perspective, and my analysis there enables the research group to further refine the mechanical understanding of this novel process. So the previous research was conducted using a molecule known as norbornene. However, my research involves dicyclopentadiene, which adds an additional fused carbon ring, which can induce an additional effect known as exo and endo. So the exo effect is when the additional fused carbon ring is in a upwards position, whereas the endo effect is when it is a downwards position. I hypothesized that the endo conformation would be less stable. So an anion, which is negatively charged, and a cation, which is positively charged, they would like to be next to each other. And with the endo ring inhibiting the anion from getting as close to the cation, it is assumed that this would result in a less stable molecule. Yes. So as shown in the previous slides, similarly, the polymer chain determines the trans versus the cis nature of the different isomers. So these two isomers are considered trans because the polymer chain is down facing, whereas these two are considered cis because the polymer chain is upwards facing. So this is a lot to take in, but what we're looking at here along the y-axis is the energy in kcals per mole, and along the x-axis is the different conformations of the isomers. What we're looking for is a strong difference in energies between the lowest energy trans molecule versus the lowest energy cis molecule. So if we look at, in orange, we have the exo conformation, meaning the ring is pointed up. The lowest energy trans molecule is exo-A, and the lowest energy cis is exo-D. So this difference is 1.6 kcals per mole. And because the trans product is lower in energy, that means that there is a trans preference indicated when DCPD exo is paired with BARF. Furthermore, when DCPD endo is paired with BARF, we see a similar trend. The lowest energy would be the endo-A conformation, the lowest energy trans would be the endo-A conformation, and the lowest energy cis would be the endo-D conformation. This is a similar difference in energy of 1.5 kcal per mole, which indicates again that trans selectivity is expected when DCPD is paired with BARF. So something of note here is that the endo molecules all exhibit a significantly higher energy than their exo counterparts. There's a strong difference between the energies in the two conformations. This is shown in this diagram here because BARF is a large molecule. When the endo ring is downwards facing, it is unable to get as close to the positively charged anion, and this difference in charge separation results in a higher energy for the molecule, meaning that it is less stable, which is reflected in the data shown. So then when the DCPD molecule is paired with BF4, there are different trends observed. So BF4 is a very small anion in comparison to BARF. It is able to get much closer to the positively charged cation than BARF can, and it is therefore presumably less impacted by the endo-exo effect of the additional fused carbon ring. So when we look at the data, there's a surprising result. The endo conformations shown in blue are in fact lower in energy than their exo counterparts. So of all the molecules, DCPD endo B has the lowest energy, which is significant because it goes against what would be assumed that the endo conformation would result in additional obstruction of the anion binding. Another really interesting point is that the exo conformation demonstrates a cis selectivity. So when we compare the lowest energy trans conformation to the lowest energy cis conformation, there's a difference of 0.9 kcals per mole favoring the cis conformation. This is significant because it shows cis selectivity. However, 0.9 kcal per mole is not as large to make it significantly cis selective, but it is of great interest and certainly cause for further analysis. So some conclusions that were observed is that the endo-exo conformation of the ring has a clear impact when paired with a large anion. The exo conformations all exhibit a lower energy. However, when DCPD was paired with BF4, which is a small anion, there was cis selectivity, and this is of great interest as the ability to select for both cis and trans is necessary in order for metal-free ROMP to be competitive against ROMP, which uses the metal catalyst. Finally, the additional fused ring in DCPD and compared to norbornene results in a swapped cis to trans selectivity with respect to anion binding. The research conducted by the Boydston Research Group indicated that with a small molecule, you would expect trans selectivity, and with a large molecule, you would expect cis selectivity. And with the additional ring in DCPD, this trend was reversed. Therefore, this is really important research to further expand the applications of the metal-free ring opening metastasis polymerization mechanism. I have a few acknowledgements. I'd of course like to thank the organizers and Dr. Mattern for moderating this event. I'd like to thank Dr. Sojin Young, Dr. Sean Gitter, and Dr. AJ Boydston at the University of Wisconsin-Madison. I would like to thank Dr. Allison Ressler for being my advisor during this project. And of course, thank you to Oglethorpe University and the University of Wisconsin-Madison. If anybody has any questions, I would be glad to answer them.
So yes, if there are any questions, please welcome people raising them at this time. I cannot see the gallery really, so do feel free. Okay, thank you, Joshua, please. And Hannah, if you can see the hands, feel free to call on people as well.
Okay, thanks.
So I see that you chose BARF as your large anion and BF4 as your small anion. Was there a reason why you chose those particularly for those purposes, or was it just one's a large anion and a small anion? And it was, I don't know, there's some consistency there.
Right. So the original research project conducted by the Boydston Research Group investigated several parameters such as solvent effects and the anion pairing and the initiator. The two anions that they focused on were BF4 and BARF, and those were the ones that they observed the greatest stereoselectivity with. So for the purposes of consistency, I used those two when conducting the research with DCPD.
Oh, Hannah, if you unshare your screen.
Oh, yes. Sorry.
If I could ask a question, as someone who's not, part of the inner circle, could you tell me about the process that's involved in undertaking how you worked through this project? I'm very curious about how one undergoes research, especially because it appears you're using various forms of research within the larger research project.
Yes, so computational chemistry is a relatively new field, considering that the computational capacity to conduct these experiments has only been achieved in the past couple of decades. So what was so first the molecules were constructed in a modeling program. They were then optimized using B3-LIP and 6-31G methods of, computational theories. So this resulted in the ideal optimized geometry of the molecule. So in nature, a molecule will aim for the most ideal conformation because it's the most stable. So the computational side of this project comes from decades of research of observing different molecules and determining what would be an optimal position. So once the molecules were in their optimized positions, the final energies were calculated and were then compared against one another. So as you may have seen in my graphs, there was always one point that had the energy 0, 0. This was because that was the reference point. When you're comparing molecules, you have to have a reference point. You can't just give just an energy because it doesn't show any significance just to give an energy. So the endo A was always the reference point, and the rest of the molecules were compared against that to determine what would be the difference in energies from those. And then it was all put into Excel and made to look pretty.
Thank you.
Yes, thank you for the questions.
Well, I have another question.
Okay.
What would be the follow-up to this? What would you wish to have happen next, both in terms of research and application?
Yeah, so I actually, so from the title of my presentation in the program, I actually conducted this research with DHDCPD, which is dihydrodicyclopentadiene. This molecule has a saturated double bond on the additional fused carbon ring. And there were actually really interesting trends that were different from what was observed with DCPD, which shows that there is some electronic impact, meaning the additional hydrogens have some impact with the anion binding. So that was, of course, really interesting, but difficult to condense for purposes of this presentation. So now that we've completed our analysis for DCPD and DHDCPD, ideally we'd like to apply this to other cyclic monomers, potentially those that have substituent groups that have electronic impacts, so something that is reactive on the additional fused ring might cause even further effects. The ideal is to refine the process and the mechanism so that it can be used for any cyclic monomer to polymerize it, and therefore replace the ring opening metastasis polymerization mechanism, which relies on a metal catalyst.
Thank you.
Yeah, thank you for the questions and thank you for having me. Oh,
I had another question if
yeah, absolutely.
So I was curious when you mentioned, so you've looked at the effect of the anion size,
Right.
and you mentioned that previous research had also looked at the solvent. So I was curious to see, like, did that have an effect, and if there are other properties or other conditions that you're interested in examining in addition to the anion size that might impact the selectivity?
Yeah, absolutely. That's a great question, and that was the paper that was published by the Boydston Research Group was very extensive in their analysis of both the initiator and the solvent effects. I believe toluene was associated with more cis selectivity, and dichloromethane was associated with more trans selectivity. That is definitely an area of further investigation. I believe that might be where my research heads going forward. Uh, so yeah, the solvent effects are definitely of interest, and the initiator was also something that was discussed by the research paper. However, that is outside of my area of expertise. Thank you.
Any other questions? Then another round of applause for Hannah. I thought fascinating. Good job, and thank you for presenting today.
Thank you for having me.
Next, let us hear about using computational protocols and empirical methods to elucidate the protein-protein interactions of MitoNEET from Joshua Amritt, who worked, I believe, with Dr. Vaughn on this research project. Take it away, Joshua.
All right, so I'm a senior here at the I'm a senior in the chemistry program here at Oglethorpe University. And as you said, I've been working on this project for a little bit under a year with Dr. Morgan Vaughn. So without further ado, this is using computational protocols and empirical methods to elucidate the protein-protein interactions of MitoNEET. There we go. So the first thing that we should probably address is what exactly is MitoNEET, that being the subject of this project. So MitoNEET belongs to a class of iron sulfur proteins, which all that's classified by is you'll notice these like yellow and orange clusters here. These are the iron sulfur clusters. They're responsible for transferring these to other proteins. This aids in a number of like biological functions, such as like nitrogen fixation, photosynthesis, cellular respiration, so on and so forth. The other aspects of this protein is the neat part of the name comes from a particular amino acid sequence that shows up later in its like overall sequence, which is 1 unit of asparagine, 2 units of glutamic acid, and 1 unit of threonine. Something else to note is it's called a neat protein because there are several other kinds. MitoNEET just happens to be the human variant, which we will be studying. And then finally, the mito part of that name comes from the fact that it's a mitochondrial protein, and it's responsible for binding to the anti-type 2 diabetes drug pioglitazone. And that's going to be the important driving factor for a lot of what we want to do with this research. The first major objective is to determine the inhibition mechanism of pioglitazone, because as I said earlier, we know that pioglitazone binds to MitoNEET, and we also know that in this relationship that it does inhibit MitoNEET. So you'll look at the, I guess, schematic diagram, whatever, that I have here, and it shows like a computationally derived image of pioglitazone binding to the MitoNEET protein. And the fact is we don't actually know where it binds. This is just computationally derived. This is probably a model that was derived using some like docking protocols, which I'll talk about a little bit later. What we would like to do is use some experiments to determine how that inhibition mechanism is actually going to happen. And our hypothesis is that pioglitazone is going to disrupt the protein-protein interactions of MitoNEET specifically. And that drives our second primary objective of this project, which is to elucidate those protein-protein interactions. And we want to look at a very specific type of protein-protein interaction, right? So because it's an iron sulfur protein, we want to look at how it's going to transfer this cluster to other proteins. For the context of this project, the partner protein that was used is going to be anamorphin, but we're going to use that to be able to monitor the holoNEET form and the apoNEET form of MitoNEET, which really is just language terms to describe when it contains the iron sulfur cluster and when it has donated that cluster. Our original goal was to monitor these things using spectroscopic techniques. We can look at complex formation with things like fluorescence spectroscopy, and we can look at actual cluster donation with things like absorbance spectroscopy. But unfortunately, due to the pandemic last year, we weren't able to do any of that. So we had to go in a completely different direction, which was utilizing computational methods, which we got a pretty deep dive in from Hannah's talk. So I don't think I'll have to go into too much detail about that. So rather, I'm just going to go ahead and tell you what programs I actually needed to use. Those being Pyroxeta, ABPBS, and QuestPro. I used Pyroxeta to work with the geometry of the protein, as well as to function as a computational middleman, if you will, because I used it to work up a bunch of files that I would be using later. The ABPBS, which stands for, I'll have it later. I'll forget for now. It's going to be used to display the electrostatic surface map so that we can better visualize the, I guess, models that we're going to get from other programs. And then I use QuestPro to actually perform the docking protocol. Because something to note is I've had no experience with computational research beforehand. So there was a lot of trial and error in wanting to use specific programs to do things, realizing that either I didn't have the expertise to make it work or it just couldn't do what I was trying to force it to do, and then having to work around that and just, develop the methods that I was using as I went along. So the first method that I want to talk to you guys about is going to be Pyroxeta. Pyroxeta is an interactive Python-based interface to the powerful Rosetta molecular modeling suite. Like I mentioned before, I'm not familiar with any computational methods before now, so I actually had to learn the program language Python to be able to use this program in the first place. But Pyroxeta is typically used to solve the protein folding problem, which you can see here on the right, this like jittery looking animation. So as we know, Hannah introduced us like monomers and polymers, right? So proteins are a type of polymer. Their monomers, or their monomeric units, are going to be amino acids, which have varying properties, ranging from electrostatic properties, polarity properties, so on and so forth. And dependent on the way they arrange themselves in their just like polymer formation, they end up folding into various particular structures, which can perform very particular functions. Unfortunately, it can be very difficult to predict that based on a given amino acid sequence. And Pyroxeta is a program that can calculate that for you, or like try and determine that for you. And it does this by simulating force fields and solvent effects on a biomolecular system and calculating the energies and surfaces of proteins. Thankfully, I didn't need to use it to do that. I used it to generate poses of proteins, calculate score functions, and to clean up the PDB files that I would use with programs like QuestPro. So with the first two parts of that, since I'm trying to look and model the protein-protein interactions, it's important that I'm able to see the geometry and energy of the proteins that I'm looking at. And the reason for this is with any chemical interaction, if you think back to the Gen Chem class, if you've taken one, if you haven't, then I guess I'm dropping this nugget of knowledge on you now. Really important things that we care about are going to be the orientation of the molecules involved in the reaction, as well as the energy that they possess, because the closer in energy that they are, and the closer they are to having the proper orientation, the more of a, like, I guess, the more of a yield you'll get from the reaction that's being run. So I used it to fulfill those two functions, as well as to clean up PDB files that I was using. A PDB file is just a file derived from the protein database, where they basically will use some sort of microscopy technique like X-ray crystallography or electron microscopy to generate a structure for a protein that's in a like crystal that's already predetermined. And then it will map this onto some XYZ coordinates that you can then use for like molecular modeling programs like this one. So I just wanted to take a second to show you what an input looked like for the pose function that I was using. This is all going to be in a terminal. As you can see, it's just, there's going to be a lot going in there. This was specifically used for a pose, which, as I mentioned before, the pose is going to be that geometric part of the interaction that I'm looking at. So this is the input, and this is the output that I get. And so what's important about this is what I get is what you see on the right, which is going to be this stuff in the terminal. It tells me some pretty important things about the geometry of the molecule, or just general information about the protein. It tells me the file name, the total number of residues, the overall sequence. I have here like specific information about residue 72. I'll revisit that. And then you'll see this one part that's like a series of L's, E's, and H's, and that tells me if a residue is a part of a helix, loop, or sheet, which are just general motifs that proteins can fold into as they're forming their tertiary structures. Finally, we have these like series of numbers down here. It's important when you're dealing with a geometry to be able to know things like dihedral angles that are present, or bond lengths, bond angles, just that kind of stuff about a geometry. This is all fine and dandy and really useful on its own, but I can't visualize any of that. If I see a sequence of amino acids, I don't know what that's going to fold into. That's kind of the reason we're using this in the first place. Thankfully, I was able to pair this with another program called PyMol. And so this is a helpful cartoon of MitoNEET, right? And it shows me those actual structures that I'm interested in. So through it, I'm able to see where like the iron sulfur clusters are located, where all those motifs show up. And then you see this highlighted portion here in red. This is that selected information about residue 72 that I was talking about. So it actually gives me a location that I can attribute all that information that I got from the output. The next part that we're going to talk about is the score function, which is going to be that energetic aspect of the interaction. The score function is calculated using a linear combination of a bunch of different forces that are acting on the protein, like within a given space. And so we're going to have like this code here that says like FA ATR, FA REP, so on and so forth. The FA just means full atom, so that respective force acting on the full atom. And you can see like with ATR and REP, we have like attractive forces, repulsive forces, solvent effects, intramolecular repulsive effects. There are like 15 more that I'm not going to go into too great of detail about, but there are a lot of different contributions to the overall energy score that we're getting. And then something to note, and I think Hannah touched on this, so I'll try to make it a little bit brief, is this energy that we get is not like is not an actual energy calculation. There's no unit attached to that, where this negative 129.72 is going to be in like kilocalories or kilojoules per mole. That's going to be a score. It's used as a relative sort of thing that you can compare to other scores that you generate. If you're using something like Pyroxeta and you're trying to generate different conformations of a given amino acid sequence, you want to have some scores to compare to be able to know which one is more or less viable, because the lower the score, the more stable that structure would suggest itself to be. And then finally, this could also be paired with PyMol. And so this MitoNEET protein that we've been looking at is colored purple because I like the color purple, but it has a distribution of energy throughout, where you can see the darker regions are dark purple and that has higher energy respectively, and the lighter regions are lower in energy respectively. So with the cartoon, that's really useful because it gives us some of that important information about how those residues are folding and what motifs they're forming. But I'm interested in looking at protein-protein interactions, which means I really need to be able to see some surface detail. And something that's really helpful is the ABPBS, which stands for the Adaptive Poisson-Boltzmann Solver. All this does is it generates an electrostatic potential map on the surface of a protein. So when you're dealing with these interactions, you're going to have the surfaces of these proteins interacting with one another in such a way that they're going to actually conform to one another. You can imagine like you have like a lock and key sort of situation going on. So that's why it's important to be able to see the surfaces, because otherwise you have something like this, where you get these models and they have their residues that sort of like line up with one another, and you can kind of tell what's interacting with what, but you don't know how those surfaces are interacting. So these models that were generated were generated by ClusPro. There we go. ClusPro is what I used to perform the actual protein-protein docking protocol. I originally wanted to use Pyroxeta, but I had to come to the realization that I don't have the computational expertise to really work with that and figure out how to use it quite yet. Ideally, I would like to, but I couldn't for now. So I wanted to consult something to be able to perform that docking for me. And thankfully, ClusPro, which is an online free server, was able to do that. So, sorry, one sec. So how it works is by a three-step process. Rigid body docking by sampling billions of conformations, root mean squared deviation-based clustering of 1,000 lowest energy structures, and refinement of selected structures using energy minimizations. What does this mean? So we have MitoNEET on the left, which is going to be the red structure, and anamorphin on the right, which is the green structure. So MitoNEET we're going to treat as the receptor here. So what they're going to do is they're going to hold MitoNEET still, and then they're going to take the ligand, which is going to be anamorphin, and they're going to rotate it billions of times and then translate it onto the surface of MitoNEET. You're going to get these billions of different just interactions that could potentially be happening between MitoNEET and anamorphin, and they're going to have a respective energy affiliated with them. Well, it's going to take the 1,000 lowest energies from those billions that you generated, and then it's going to refine them further using energy minimizations to give you a final calculated score. So with that, you could see that the residues were hard to like, see where the interactions were happening. So I used the ABPBS to overlay onto those structures so that I could actually see the surface detail of what was going on. So some of the structures that I have here, or really all of them, I parsed through the models that I got out of PlusPro, and I took the lowest energy ones, and then I also took the interactions that showed anamorphin being close enough in proximity to the iron sulfur cluster of MitoNEET. And then I used that to kind of generate some structures that I felt would best elucidate the actual iron sulfur cluster transfer mechanism, right? So in this first one that we're looking at, you'll see that you have this electronegative region of anamorphin. By the way, electronegative regions are in red, electropositive regions are in blue. I took this electronegative region of anamorphin, and you can see that it almost fits perfectly in a little pocket of MitoNEET, like right here. And I thought that was really cool because that helps to explain why it would have such a stable energy affiliated with it. They have opposing electrostatic interactions, and one practically fits into the mold of the other, which is really cool. And then with the image on the right, I just wanted to be able to better show the proximity of anamorphin specifically to the iron sulfur cluster. Because again, I really have to reiterate, what we care about is seeing that iron sulfur cluster transfer. We don't want to see just any sort of interaction that just, looks like it could work. With this next structure that I have, you're going to see the same sort of tune carried across with a lot of these structures where you have the opposing electrostatic interactions going on. But what I thought was particularly interesting about this interaction is if you look at the image on the right, you have this like divot in the anamorphin structure, or like a concave region or whatever. And that's where the iron sulfur cluster actually happens to be located, relevant to that. And so I thought that was interesting because you can imagine that you could now have that cluster transfer happening like internally, which would be a lot more stable of a transfer, I would imagine, than something where it has to transfer it into like the cytoplasm, if that makes sense, where that's happening outside of the two proteins. So this would be a good way to facilitate that transfer. I have a couple other models here. Same tune again, anamorphin is close to the iron sulfur cluster. Opposing electrostatic interactions. I liked the bottom three here because in the other structures, you kind of had like one region of the protein that had opposing electrostatic interactions with the other, and it was a very like small, concise, distinct region. But with this particular model on the bottom, it was almost like all of the structure was properly aligned with the opposing like electrostatics of the other structure, and they were just like perfectly puzzle fit together. It was really cool. So I wanted to show multiple angles of that just to, better display that. So with this having been done, what are the conclusions I was able to draw, and what are our plans for the future? The first thing that we want to do is determine the anamorphin structure with the iron sulfur cluster. The reason why this is important is you can imagine if we want to see an iron cluster transfer, then obviously we have the MitoNEET that has the iron clusters to begin with, which, as we said earlier, that's going to be the hollow NEET form. And then we want to be able to have a mechanism that shows the transfer of that iron sulfur cluster to anamorphin. And then the last part of that, like the last puzzle piece there, would be anamorphin actually having the iron sulfur cluster, because that would demonstrate that a transfer has occurred. So I would want to be able to find a structure that exists for that so that we could better actually model that sort of mechanism. The next thing is we want to perform the docking simulations with pioglitazone. That's going to be really, really important because one of our major research objectives was to see that pioglitazone was inhibiting this actual protein-protein interaction. Not necessarily what I'm hoping to see, but what I'd be interested in seeing is knowing that it inhibits MitoNEET, I'd be interested to see if it overall raises the energy functions or if it sort of changes the way that they will interact with, like geometrically speaking, where we might see less of a sort of overlap between the iron sulfur cluster region of MitoNEET and anamorphin. So we just got to introduce pioglitazone to further elucidate that. And then other things we want to do is determine which residues in both proteins are responsible for the binding interactions. that's technically something we can actually do already, but it would require some very involved looking through the PDB files that I got out of QuestPro, because I would have to see where the coordinates of the atoms actually happen to be in really close enough proximity to each other for that. And you can imagine there are a lot of atoms in these proteins. That's a lot of data to have to look through, and I didn't want to do that quite yet. So we do have to do that at some point, though. Something that I wanted to point out, just because it's an important note about Pyroxeta, is that it is not deterministic. One of the amazing benefits of computational research is that you can do it from your own computer, but there is a cost that comes with that, especially if the computer you happen to have is not a super expensive or powerful one. And that is that the calculations that you get out are not necessarily going to be the most accurate. So they really stress that when you're using Pyroxeta, because it's the light version of Rosetta, that you take as many trajectories as possible as you can. And that's just something that I wanted to make clear, so that like when you saw whatever data I had, that was a result of me taking quite a few trajectories, but it's still something where I wanted to make that clear. And then finally, due to us being able to return to campus, like with this most recent semester, I have been able to actually do like the wet lab portions of this experiment so that we can get on track to do the spectroscopic experimentation techniques that we wanted to do. And so what's cool about that is we'll be able to gather empirical data that we can then use to compare to the computational data that we got. And we'll be able to find sort of like an overlay between the two. And I'm currently actually finishing up the protein expression and purification part of that so that we can start to move on to working with our protein to begin with. With that being said, do I have any questions? Or do you have any questions? There we go. Not me having. Okay. I'm going to stop sharing now.
First, let me say beautifully timed. I didn't even flash any colors at you because I could see that you were right on track. And yes, I'd love to open the floor to questions.
I guess I'm going to take these as I see them. So Hannah.
Okay, so I have several questions, so I'll just ask one and then let somebody else ask another. I was curious about what your wet lab involvement entails. If you could talk a bit more about what you have just completed now that you're back.
Okay, so yeah, I'm currently doing the protein like transformation, expression, and purification, which all that really means is I'm making me some protein. I'm using bacteria to do it. And so it involves taking like a plasmid, putting that in an E. coli bacteria, making many of that bacteria, and then forcing that bacteria to make me the protein that I want, and then bursting open those actual cells to farm me some of that protein so that I can then proceed to the spectroscopic aspects of this experiment.
Wow.
That was a lot more violent of a way to describe it, but it's the way I think about it, and it makes it fun. Yes, Dr. Handler?
Hello. I don't have a question, but I just wanted to say that you are a very polished and entertaining presenter.
Thank you. I think Anissa had a question. And went up and went down, I don't know.
Oh, no, I didn't have a question now.
Okay, okay. Did you have a question, Dr. Ressler? I don't know if I saw your hand go up. Like 3 of them went up at once and then all 3 of them went down at once. So I only remember seeing Hannah's. Okay. Well, any other questions?
It's like Taylor's got her hand up.
Is there a second page? All right. Well, Taylor, what's your question? I can't.
Hey, so I was wanting to know, besides fluorescence, are you guys going to try to use any other spectroscopic techniques to look at your proteins?
So we will be using absorbance techniques because the actual iron sulfur cluster will, I don't know, it'll provide an absorbance reading, and we'll be able to see how that concentration changes over time with the amount of absorbance that's happening, right? So if we see an inhibition of that, then we can imagine that that change in absorbance over time will change as well. There we go. So yes, there will be other spectroscopic methods involved. Hannah, other question of many.
Yes, hopefully not too many. I was curious about the functional purpose of MitoNEET in a human.
That's a good question. I honestly don't know. Like I mentioned, it is involved in performing like a lot of the key biological functions like cellular respiration and
Oh,
so forth. But in terms of the very specific way that it is involved in that, I do not know. Dr. Hirschler?
Yes, fine. My question is a meta question, which you probably, there may not be an answer to because it involves working with counterfactuals. So the idea of listening to the way you described your project, and I really appreciate the narrative that you provided to go along with how you were gathering and working with the data. You know, the question that everyone's asking, how has the pandemic affected your education? And therefore, I'm curious, it sounds like while there have been challenges, it's forced you to do things and learn things that you wouldn't necessarily have learned otherwise. So can you reflect upon how going through this research, doing this project, how it's been impacted by the pandemic and what that suggests about the study of chemistry for you now and going forward?
So something to note is, I'm not gonna lie, the main reason I wanted to do research this semester is because I was applying for grad schools and I wanted to make sure I had a solid research foundation base, you know, to put on my resume. And so I guess being a chemistry major, I should have been doing that anyway, but that was something I wanted to make sure I had in addition. And so because of that, I already was going in with the attitude of, I don't care what situation we're in, I've got to figure out a way to get some research background going. And so with that in mind, I think the general attitude that you're supposed to have when you're working with research is you're going to run into walls, you're going to run into some things that it's going to force you to think outside the box and figure out, how am I going to deal with this wall so that I can continue whatever I'm researching. And what was cool about this experience is that didn't necessarily go away. That was something where, even though it was being done virtually and mostly computationally, I still had a very similar research experience to what I guess I was expecting to have, where I would have to make sure I figured out a way to gather data, make sure I had to figure out how to communicate that data. I was regularly speaking with Dr. Vaughn to go over whatever I had been working on, kind of like research meetings slash, what's the term? I guess making sure I'm just doing what I'm supposed to do type meetings. Forget the word for that right now. But all of this experience was still something that I was able to do. And that was due in part because, I don't know, just like with any attitude you have when you're doing research, you just got to keep pushing and you got to keep doing it. So that being said, I'm willing to say that I didn't, it's not that I didn't learn anything that I wouldn't have learned if I didn't do computational research, or, you know, like that the pandemic, I don't know, made me learn anything different. But I think it was one of those things where I definitely felt like I had to work harder to make sure that I could still accomplish what it is that I wanted to do.
Great, thank you.
Hannah, yet again.
yes. Okay, last one. And also, I just want to say you definitely did accomplish plenty. So kudos to you.
Thank you.
I was curious about your Python input file. So just scanning what you showed on the screen, it was really interesting to see the methods that were used. Did you feel that you were able to, you said you didn't have experience with computer science beforehand. Was it something that you were able to sort of follow the manuals and parse the language? Because a lot of the methods were like hyper specific to the program. I was just curious if it was like you made a text file with all your commands and then loaded it into the terminal in some way. I was just curious about the process itself.
Okay, so I guess a way to put into perspective how little experience I've had with this, I spent maybe 2 months struggling to like just download the thing, like not even like starting to use it, just downloading it onto my computer so that I could use it. It took me practically crying to one of my friends, like, hey, I'm trying to use this thing and I don't know how to use it, and you're a computer science major, you will help me. And he did. And once we got that out of the way, he was like, Josh, have you considered actually learning the programming language so that you could do this? And I was like, wow, that never clicked for me. No. And then he basically pointed me in the direction of like different Python tutorials that were available online that were free, and there are plenty. And so that was the first thing I had to do. From there, you're right. There were a lot of hyper-specific commands that went in. I actually basically took advantage of a lot of the workshops that they had online for this particular program. And so one of the workshops may have been focusing on how to manipulate the pose of your protein. I was able to take advantage of that. And so what I would do is I would take a workshop, I would break it down into literally every single command that was there. I would figure out how to make that command apply to the protein that I was interested in, which in this case happened to be MitoNEET. And then I would write a script that contained all of that. And I would press enter, see if it ran. If I ran into any issues, I would figure out which line specifically caused that issue. And then I would work from there to hopefully overall generate a script that worked entirely.
Josh, I just wanted to say that I think it's really impressive what you accomplished over this past year. And if it makes you feel any better, I think that downloading and installing computational software is the hardest part of a research project. So if you end up doing any computations in grad school, I hope that you'll be part of a group that hires someone to do all of that for you.
I do too. I can't stress how frustrating that was. Like, I don't know. I was looking at the workshops for how to do a lot of the things. Like, for example, I wanted to do the docking protocol. I ran into an error there that I didn't know how to fix. But for the most part, it was working. Like, there were a lot of aspects of it that were working. And I remember thinking, why is it this isn't that bad compared to just downloading the thing? Why couldn't they make that easier? I don't know. Like, I just felt like that needed a workshop in and of itself, and they just didn't have one. And that really confused me.
Yeah,
But
that's how it always is, unfortunately.
Excellent, folks. Well, if there are no more questions, one more round of applause, please, for Joshua. Two fascinating, really impressive presentations. And let us now move on to another form of creativity, a little less chemical, but with a chemistry of its own. So now we're going to hear, yes, sorry about that. A handful of pills, through lines and abiding images in creative writing by Ivy Jones and Kimberly Farmer.
Right, on a completely different note.
Okay.
All right.
So, hi, I'm Ivy Jones. This is Kimberly Farmer, and we're presenting on Through Lines and Abiding Images in Creative Writing, based on work we did in Writing 490 Advanced Special Topics in Writing under Professor Jessica Handler.
So before we start, as has been the trend so far, let's define some important terms. A through line is a consistent metaphor that holds a piece of creative writing together. It's basically the message of the story, no matter what the actual plot line ends up being. The abiding image is the representation of that through line in a specific form. So because the abiding image inherently represents that through line, the author chooses these two deliberately in order to form the creative vision of their work. So if we look at The Great Gatsby, which is a book that most people are familiar with, we can identify the through line as hope and the American dream, and then the abiding image that then represents that through line as the green light that Gatsby references all the time. As we can see, at least in this quote, Gatsby believed in the green light, the orgastic future that year by year recedes before us. So we can see both terms occurring concurrently. Look at that. Similarly, in our Writing 490 class, we as authors had to deliberately choose our stories through lines and abiding images. So let us introduce you to the stories we've been working on.
My project was St. Clotilde's Home for Troubled Youth, a novel in verse. As the title says, it's a verse novel, which for those who don't know is kind of a hybrid form between a poetry collection and a novel. It's generally a series of poems that carry a narrative and novel or novella length in publication. It's set in a surreal boarding school group home, and it follows the protagonist Suki Posey through her involuntary stay at this group home and she interacts with characters such as Bella and Leon Lovebird, who are teachers' pets, Miriam, a girl who speaks in couplets, Harmon, a ghost-like familiar figure who haunts the hallways, and Turquoise, a little boy who lives in an upstairs bathroom and becomes an unlikely friend. The format of my verse novel is that it's split into 5 parts, which is introduction songs, and then parts 1, 2, and 3, and Finn. And inside those parts are individual poems, and there's like 10 or so individual poems per section, like with differing lengths. My through line is human connection and how it can be found against all odds. This like focuses on, in the narrative, as Suki's struggle of how she wants to avoid human connection, but she finds it anyway. The abiding image is hands. And I chose that image because hands represent touch and connection physically between people. And I wanted to bring that image in as a metaphor in my verse novel to play with tying together the narrative through the sinister and trustworthy nature of the image of hands. So this is a section from, this is the first poem in the novel, called The Little Boy and the Headmistress. They check out his teeth with turquoise fingers. This is not the oddest thing. He opens wide and lets them peer into his mouth. He's a bit sick in the head. The fingers retract and he shuts his mouth. It's the porcelain of the sink digging into his spine and ribs. It feels like a butter knife at his back. It feels like a public bathroom sink. The woman clicks her tongue, shakes her head as if to free an unpleasant thought. He is having unpleasant thoughts. He may be anxious. He's a bit sick in the head. The sun filters through the watery paned windows that hang on the far wall. The frames of the windows are holding the glass back from an attack almost. His teeth are perfect. This is particularly odd. He eats lots of sweets and his molars sometimes ache in his jaw, foreign objects, pearls in his mouth, turquoise on his tongue. The woman steps back finally, nods to the 2 others. The girl's hand is braced on her hip, the other arm lank against her side. Her fingers are human fingers. They have not pried his mouth open to investigate. The other leans against the bathroom stall divider board. The woman steps back, the boy steps forward. His feet feel achy on the tile. Her name ends up being Madame Amaryllis. His ends up being Turquoise. So we've highlighted the sections where you can see my abiding image here with like fingers mainly and hands. And then you can also see the through line in these yellow boxes of like community and like working together to achieve a goal. And this next selection is a selection from part 2, poem 8, where Suki is having a fever hallucination. The wrist connects to the hand as a knob piece, same as door, and then come the bones. They are called bones. They end in joint knuckles, extend fingers, end in fingernails, tips of bone, tips of skin, back down, muscle and vein, the alabaster chalk prints of delicate body parts. Then comes the thumb, pointer, middle, ring, pinky. Then comes the red ones. Then comes the palms, where the world is mapped out on a dinner plate. And so that is like basically all an abiding image, which I just wanted to reference for just part of my story that doesn't have any through line in it, but just abiding image because it's poetry.
Right, so next up, my story that I've been working on is called Refuge so far as my tentative title. It's a prose novel, which is a fancy word for it's written like a regular novel instead of in verse like a poem. I've called it Refuge right now because it's set in a boarding school that functions as an idealized form of an individualized education plan for kids who are struggling in school. The school individually caters to each student's needs and therefore acts as a refuge for them. So we follow my main character, psychiatric nurse Milo, as he counsels these students to the best of his ability, even when they turn out to be superpowered, and therefore these things that are keeping them from doing well in school may actually be a side effect of their superpowers after all. Hopefully, after I get this all done, I might end up turning this into a series. So my through line has been control, which usually shows up in the form of regaining control over one's own life. And my abiding image is prescription medication and treatment plans. And I chose that image because usually in the case of mental illness, the severity of that mental illness tends to be assessed by the lack of control you have over your life, or how badly that mental illness affects your daily happenings. And prescription medication is usually the first line of defense in regaining that control. Milo, my main character, a nurse, is notoriously open about finding new ways to help his patients, including switching medications when they don't seem to work or trying new things. So my first example is from a talk with one of my kids, Callista. And let me Read it out. It's been 3 weeks, she finally burst out, rocking forward in her precarious seat. You said the meds should kick in 2. No change? Milo's eyebrows shot up in surprise. At all? A sharp shake of the head. Left, right. Have you been trying those breathing techniques we talked about? He continued, hoping for at least a little reprieve from her usual cautious nature. His wish came true in the form of a much smaller, less certain nod. Do they make any difference? A shrug. Scrunch, release. Milo held in the sigh that threatened to slip loose. Well, if the meds aren't making a dent in things, we'll scrap them. But if the breathing techniques are making any difference, keep it up, all right? He encouraged her with a smile. Besides, the symptoms you're experiencing may not have anything to do with anxiety after all. It could very well be a side effect of your powers. So here in my first example, we can see my abiding image of prescription medications and how they're not working. Then in my through line, we can see the regain of that control through alternative methods, methods that may not have been prescribed initially, but are more effective than those medications. Then we move on to a section with Jordan. Did you need something before I leave? Jordan asked with a tilt of her head, one hand halfway to reaching for the bag at her feet. Yeah, just one question, Milo said as he struggled to find the right words. Has your outlook on life changed any since coming here? For a moment, he didn't think Jordan would understand what he was asking, but to his surprise, her entire face lit up in the brightest smile he'd ever seen her wear. Yeah, she chirped, beaming at him. Things are starting to make sense now that I actually know what's going on with me. I'm not just an unsolvable case for doctors to puzzle over anymore. I can actually fight back against the stuff standing in the way of my everyday life. Things are way better than before. So here my abiding image can be seen through Jordan's self-awareness by way of her treatment so far, and how she actually knows what's going on. And my through line of control comes through in the improvement of her life through that treatment. So you can see in both of our works, the use and placement of through lines and abiding images is deliberate, and it rarely appears for no reason at all.
So in conclusion, creative writing is a diverse field full of works that invite a deeper analytical consideration. This is because some through lines and abiding images are obvious and easy to point out, while others are so well woven into the story that their significance is not immediately clear. As we've demonstrated through examples of published and personal works, through lines and abiding images are connected. Together, they serve to further illustrate the purpose of the story as a whole.
And now you understand why our presentation is called A Handful of Pills.
It's not just random.
Right. We're not crazy, we swear. That was on purpose.
Great, thank you so much. And also wonderful to have this, true example of direct creativity in the form of writing. If there are questions, let us open the floor to them. It looks like we have one already.
Yeah, go ahead.
There are 2 Joshes, each with the hand up. I wasn't sure which one you wanted, so I'm going to go first. I guess, how do you go about determining your abiding image / through line, right? Like, I guess, what kind of inspires you to do that?
So,
oh, you go first.
Who wants? I'll go first. So the through line is usually something you have in mind when you start writing it, because you write your story, but you also write your story. And what like with how like Aesop's fables were with like, it's a story, but it means this. And that's usually your through line. The abiding image is a little harder. Sometimes you're like, yeah, I know what metaphor I'm going to use to represent my theme. And sometimes you're like, I really got to think about this. I personally was like, I don't know. I had to like talk the whole story over with my friend before going, it's right in front of my face. It has been there the whole time.
Yeah, for me, I get sections. My mind goes ahead and does it for me a lot of the time. It's just like, you want to work on this to like represent this. So I'm like, I read over sections I've already written and I'm like, oh, cool. It's done for me.
Thank you.
Hannah?
So first of all, great presentation. I really love the creative writing styles that both of you use. I found all the passages that you shared really engaging and interesting. I was very curious that both of you had your pieces placed in a school setting. I was wondering if that was a coincidence or if that was the prompt from class.
Yeah, it was a coincidence.
It was a coincidence.
We both were like, oh, look, both of our stories are like homes for troubled children.
Yeah, it's like, oops.
Is that what made you two decide to join together for a LASS presentation? Or I was just curious how you decided to.
No.
We
It
kind of-
was like the coincidence of us like
Yeah.
both doing it and both wanting to do LASS.
Mhm.
Very cool. Yeah, really interesting presentation. I learned a lot.
Thank you. Leah?
Can you have more than one through line in a story?
Yes.
I think so. I think there's always going to be a main through line. It's like when you read a book, there's more than one theme going on at once. Like there's obviously the main what's going on, the main meaning of the story, but there's also the little things that go on too. So I think in the definition of through line's the main one, but you can have different like little through lines or little abiding images that like go off of that, I guess.
I have a question. Nice job, y'all. Kimberly, you started to talk about this with Aesop, but how does through line or abiding image connect with the idea in creative writing of what we talk about, the thing and the other thing? Can you kind of make that connection?
So I would say that your through line, wait, you cut out. What was that last part?
Oh no.
Either of you or both of you.
Oh, okay.
Saying is you started to talk about it with the reference to Aesop, but how does the through line and the manifestation through the abiding image support the idea of, the thing and the other thing in writing?
Yes. I would say, and correct me if I'm wrong, that your through line is usually the other thing. That and that's varying levels of clear depending on how convoluted you write your story. We've talked about in class how like some stories are like, that is the theme, that is the other thing that we're looking for here. Like they're talking, or like, for example, with Zootopia, the movie, it's like they're talking about animals, but actually they're talking about classism and racism. But like, if you were just like looking on a surface level, you'd be like, that's a bunny in a box, really cool.
Mm-hmm.
But then, in some other pieces, you read over it and you're like, okay, if that had a deeper meaning, I didn't catch it. I thought it was about, I thought the curtains were just yellow. I didn't know that they had a meaning.
Very nice, thank you.
I have a question. So I really enjoy it, and I really appreciate hearing students' creative works. That's a great treat for me. One of my areas of specialization is metaphor. I actually go to conferences in various countries and listen to and present on metaphor. It's multidisciplinary, so I'm no expert. It's all over the place. Metaphor is a monster, but I'm interested in the definition. There you go. But I'm interested in the definition of the through line being a consistent metaphor, and therefore I'm just asking if you could elaborate a bit on the through line as metaphor. If there's somewhere you can take that. It's interesting to have something that moves through something, and yet at the same time as a metaphor suggests kind of structural properties that are not temporal, but kind of structural, if that makes sense.
Yes. Do you mean like the abiding image metaphor?
I guess you said the through line in a creative writing narrative is the consistent metaphor.
Oh, yeah.
That
Okay.
holds the piece together.
Yes. So using that, Ivy, do you want to take this one? Because I can like spout off, but
You go ahead, spout off.
well, okay, I'll spout off. All right. So like using that consistent metaphor, and often when you combine that with the plot, like you said, with like it being a structural thing, like your metaphor moves with your story. So like, you know that like a plot is going to have the rising action and the climax and the falling action and the resolution, where like the character has to change at some point, or else why are you reading the story? Because the character never changed. So your through line often moves with that, where like in Ivy's case, it starts out as like, I don't want any human connection. I'm not connecting with anyone. But by the end of the story, it's like, I have made this human connection and it has changed me as a person. And where in mine, it starts out as like, these students are like, I have no control over my life. I am adrift in the ocean of whatever is happening to me. And by the end, they're like, okay, I have a little bit more control with the threat of whatever's trying to take that control away. And then Milo himself being like, do I have any control over any of this? And having to grapple with that through your story as the plot moves.
And so in some respects, the metaphors, it's a metaphor, but it's how it's played and how it manifests changes over the course, the arc of the narrative. Okay,
Yes.
Thank you. Very interesting.
Joshua?
Yes. So I guess when you have an established, I guess, through line or abiding image that you're working with, I guess, how do you navigate just as a creative writer, avoiding any cliches affiliated with that particular abiding image that you're working with?
Well, I don't know how to phrase this. It's easier to know what the cliches you have and like need to avoid are, and it's easier to fill them in a place where they could be, and then just completely take apart the cliche and move it around so that you still have the same effect. But like, it's like, I'm trying to think, I can't think of a good cliche off the top of my head that has to do with my abiding image. But no, wait, yeah, okay. So like, you know, when people write and somebody's had their fist clenched and they like, you open your hand and it says the little half moons on their palm. I forgot what I did for that.
But I remember you doing something
did something
specifically
for that.
different, and I was like, that really stood out to me because I have read crescent moons over and over and over again.
Yeah.
You know?
Yeah. So basically, I think it's just a matter of understanding which are cliches and what you need to look at to like get your mind going to avoid that, I guess.
And in the end, there's only 88 keys on a piano. You're gonna run into some things that you're just gonna have to use.
Mm-hmm.
As long as your whole story isn't just the same key over and over again, then you're good.
Kind of experimental though.
Well, if you're broadly educated in the liberal arts, you're going to have much more to draw upon. So you're less likely to fall into those traps. So do we have other questions? See no hands. Let us one more time give a round of applause to all our presenters. Really wonderful, really impressive. Much appreciated. This session is being recorded, which means it should be available for everybody at some point thereafter. I thank you all for coming today. I thank the presenters for their contributions. I thank the audience for their attention and their questions. And I wish everybody well. And don't forget, LASS is continuing today and tomorrow with various readings, performances. PRISM is happening. Please go out, engage, celebrate. Thank you.